Hello, my name is Sechelle King, and today we will be talking about genetics and Mendelian concepts. Okay, let's go over genotype. So, genotype refers to the specific genetic makeup or combination of genes that an individual possesses. Genotype is determined by the combination of alleles, which are different versions of genes. Genotype plays a crucial role in determining an individual's phenotype, which is the observable characteristics of traits. For example, eye color, height. These are influenced by genes. While some traits are directly linked to genotype, like blood, others are influenced by multiple genes and environmental factors, making the genotype-phenotype relationship more complex. Genotype information can be obtained through genetic testing, which is used in medical genetics, forensics, and research to understand genetic predispositions and hereditary conditions. Let's talk about the importance of genetics in research. So, genotype information is fundamental in genetics research to study inheritance patterns, genetic diseases, and the molecular traits of these diseases. So, to summarize, genotype represents an individual's unique genetic makeup, determined by the combination of alleles inherited from parents, and it plays a central role in genetics research and understanding hereditary traits. Now let's talk about phenotype. Phenotype refers to the observable physical and biochemical characteristics of an organism, which results from the interaction between an individual's genetic makeup, their genotype, and environmental influences. So let's talk about external traits. Phenotype encompasses all external traits that can be seen, measured, or otherwise observed, including physical features like eye color and height, and biochemical traits like blood type. So let's talk about genotype and phenotype relationships. Phenotype is the result of the expression of an individual's genotype. It reflects how an organism's genes are translated into traits. Influence of genes. So genes play a significant role in determining an organism's phenotype by providing the instructions for the development and functioning of these various traits. And let's touch on environmental factors. So phenotype is not solely determined by genes. Environmental factors such as nutrition, exposure to toxins, and temperatures can also influence phenotype. This interaction between genes and the environment is known as genotype-environment interaction. So let's talk about phenotype and its study in genetics. Phenotypic analysis is a crucial aspect of genetics research, as it allows scientists to explore the effects of genetic mutations, study inheritance patterns, and understand the molecular bias of these traits. And let's touch on phenotypic plasticity. So some organisms exhibit phenotypic plasticity, the ability to adjust their phenotype in response to environmental changes. For example, plants may grow differently in response to varying light conditions. So to summarize, phenotype encompasses all observable traits of an organism, reflecting the interplay between an individual's genetic makeup and the environmental influences. It is a fundamental concept in genetics and essential for understanding how genes and the environment shape an organism's characteristics. So on this slide, we will be talking about epigenetics. So epigenetics is the study of changes in gene expression or cellular phenotype that do not involve alterations to the underlying DNA sequence. Instead, epigenetic modifications involve chemical modifications to the DNA molecule or changes in the structure of chromatin, which is the material that makes up chromosomes. These modifications can be heritable and can influence how genes are turned on and off. So let's go over some key concepts in epigenetics. So let's talk about DNA methylation. So methylation is an addition of a methyl group to the DNA molecule. Methylation often results in the repression of gene expression. It can be passed from one generation of cells to the next during cell division, making it a key mechanism of epigenetic inheritance. Now let's talk about histone modifications. Histones are proteins that package and condense DNA into chromatin. Chemical modifications to histones, such as acetylation or methylation, can alter the structure of chromatin, making genes more or less accessible for transcription. Now let's talk about non-coding RNAs. So, certain RNA molecules, such as microRNAs and long non-coding RNAs, can influence gene expression by binding to mRNAs and preventing their translation into proteins or by affecting chromatin structure. Then, of course, we have our environmental influences. So, external factors, including diet, stress, toxins, and exposure to certain chemicals or drugs, can induce epigenetic changes. These changes can potentially impact health and disease risk. Development and differentiation. Epigenetic modifications play a crucial role in the development of multicellular organisms. They help determine which genes are active or silent in specific cell types, leading to cell differentiation. So let's also touch on the disease and health aspect of epigenetics. Epigenetic changes have been linked to various diseases, including cancer, autoimmune disorders, and neurological conditions. If we can understand epigenetics, then we can provide insights into disease mechanisms and potential therapeutic interventions. So let's talk about the reversibility of this. So, unlike changes in DNA sequence, epigenetic modifications are reversible. This reversibility offers potential avenues for epigenetic therapies to treat or manage diseases. So, to summarize, epigenetics explores the heritable changes in gene expression that occur with alterations to the DNA sequence. It is a dynamic and evolving field with significant implications for understanding development, disease, and potential therapeutic interventions. Next, let's talk about Mendel's experimental design. So his choice of organism was the garden pea plant. He chose this for his experiments due to its ease of cultivation, short generation time, and ability to self-fertilize or cross-fertilize. So he began with isolation of the purebred lines. He started with purebred pea plants that consistently exhibited specific traits such as tall or short. He then controlled the cross-pollination. To control the breeding process, Mendel manually cross-pollinated these pea plants. Next, he was generation tracking. He tracked the traits of the parental generation and their offspring, the F1 generation, through multiple generations. Observation of traits. He observed the presence or absence of specific traits such as tall or short in each generation from these pea plants. He then collected quantitative data. Mendel counted the number of offspring expressing each trait to obtain this data. Then he performed statistical analysis. He used this to derive ratios and patterns from the data. So he would get like short or tall, yellow or green, wrinkled or smooth seed textures. From his experiment, we were brought the law of segregation and the law of independent assortment. The law of segregation states that alleles or gene variants segregate or separate during gamete formation, ensuring each offspring inherits one allele from each parent. The law of independent assortment states alleles for different traits segregate independently of each other during gamete formation, leading to various combinations of traits in offspring. Mendel's groundbreaking work was published in 1866. Though it initially received limited attention, his discoveries laid the foundation for modern genetics. Mendel's experimental design, with its focus on controlled breeding, systematic observation, and quantitative analysis, provided the empirical basis for our understanding of the principle of inheritance, now known as Mendelian genetics. Here I included this photo so we can look at different traits from these pea plants. So tall versus short, the different colors versus the different textures of the seeds. Now let's talk about monohybrid crosses. A monohybrid cross is a genetic experiment that focuses on the inheritance of a single trait or characteristic. In this type of cross, two individuals that differ in one specific trait are bred to understand how that trait is passed from one generation to the next. So let's go over the monohybrid cross step by step. So we will select a parental generation. This will be coded as capital P. So then we'll choose two parent organisms with different alleles for a single trait. For example, let's consider flower color in pea plants. One parent with a dominant allele for purple, we'll call that capital P, or the other with recessive allele for white flowers, that will be lowercase p, lowercase p. Number two, we will determine the genotypes of parents. So the parent with the purple flowers has a genotype of capital P, so that will be two dominant alleles. And the one with the white flowers has a genotype of two lowercase p's, that's the two recessive alleles. Then we would cross the parents. We'll cross the two selected parents. Each parent contributes one allele to their offspring. Number four, we'll observe the first generation, so the F1 generation. The offspring of the F1 generation will all have a genotype of capital P, lowercase p. So they would have one dominant allele from one parent and one recessive allele from the other. In this case, they will all have purple flowers because the dominant allele, capital P, masks the recessive allele, lowercase p, in determining flower color. If we were to move on and observe the second generation, the F2 generation, this generation we'd see a 3-to-1 ratio of capital P, or capital P, lowercase p, to white flowers, the lowercase p flowers. So to conclude, the monohybrid cross demonstrates how a single trait, in this case flower color, is inherited from one generation to the next. It illustrates Mendel's law of segregation, which states that alleles segregate during gamete formation and are randomly combined in offspring. The dominant allele, capital P, masks the effect of the recessive allele, lowercase p, in determining the traits. Now we will talk about a dihybrid cross. So the steps in a dihybrid cross would be selecting a parental generation. So you choose two parent organisms with different alleles for two distinct traits. For example, we can consider pea plants again with flower color and seed texture. One parent would have purple flowers, capital P, and smooth seeds, capital S. The other parent would have white flowers, lowercase p's, and wrinkled seeds, two lowercase s's. Then we'd have to determine the genotype of the parents. The first parent would have a genotype of capital P's and capital S's, two dominant alleles for the flower color and seed texture, while the second parent has a genotype of two lowercase p's and two lowercase s's, two recessive alleles. We would then have to cross the parents. We'd cross the two selected parents. Each parent contributes one allele for each trait to their offspring. Then we would observe the F1 generation. So the F1 generation will all have the genotype of capital P, lowercase p, capital S, lowercase s, one dominant allele and one recessive allele for each trait. In this case, they will all have purple flowers due to the dominant allele for the flower color being capital P, and smooth seeds due to the dominant allele for the seed texture, capital S. If we were to move on and observe the F2 generation, we would see a different ratio. 9 out of 9/16 would have purple flowers and smooth seeds, 3/16 would have purple flowers and wrinkled seeds, another 3/16 would have white flowers and smooth seeds, and 1 out of 16 would have white flowers and wrinkled seeds. To conclude, a dihybrid cross demonstrates how different traits are inherited together from one generation to the next. It illustrates Mendel's law of independent assortment, which states that alleles from different traits segregate independently of each other during gamete formation and are randomly combined in offspring. This genetic experiment helps us to understand how genes for multiple traits are inherited and how different combinations of alleles can result in various phenotypic outcomes in offspring. Now let's talk about neo-Darwinian synthesis, also known as the modern synthesis. This is a major theoretical framework in evolutionary biology that combines Charles Darwin's theory of natural selection with Mendel's principles of genetics. It emerged in the early 20th century and has been influential in shaping our understanding of how evolution works. Let's touch on the key elements of this synthesis. So number 1 would be natural selection. This concept is proposed by Charles Darwin in the 19th century. This suggests that individuals with advantageous traits are more likely to survive and reproduce, passing those traits to the next generation. Natural selection is a key driver of evolution. Number 2 would be genetics. Mendel's work in the late 19th century established the principles of inheritance, including the concept of discrete genetic units, also known as genes, and the rules of heredity. Number 3 would be mutations. Neo-Darwinism incorporates the idea that mutations, which are changes in genetic material, are a source of genetic variation. Mutations provide the raw material upon which natural selection acts. To continue off our last slide, we'll talk about number 4, population genetics. The neo-Darwinian synthesis introduced the field of population genetics, which studies the genetic composition of populations over time. It combines Mendelian genetics with the principles of natural selection to explain how genetic variation is maintained and how the new traits spread through populations. Number 5 would be modern molecular bio. Advances in molecular biology, such as DNA sequencing and the discovery of the genetic code, have reinforced and expanded the neo-Darwinian framework. They allow us to study genes, mutations, and evolutionary relationships at the molecular level. Now let's talk about microevolution and macroevolution. The neo-Darwinian synthesis addresses both microevolution, changes within a population, and macroevolution, the origin of new species, by explaining how gradual changes at the genetic level can lead to speciation and diversification of life. Now let's talk about the synthesis of these ideas. The neo-Darwinian synthesis integrated Darwin's concept of natural selection with Mendelian genetics and laid the foundation for the modern understanding of evolution. It provided a comprehensive framework to explain how genetic variation and natural selection and inheritance collectively drive the evolution process. So to summarize, the neo-Darwinian synthesis is a crucial framework that combines Darwin's theories and Mendelian genetics, population genetics, and modern molecular biology. It forms the cornerstone of our current understanding of how genetic variation and selection drive the process of evolution. Now let's talk about the chi-square test. So the chi-square test is a statistical tool used to check if two categorical variables are related or independent. This test is a common approach to assess whether observed genetic data deviates from expected proportions. This test is often used in genetics to determine if observed genetic ratios, such as Mendelian inheritance patterns, or they're also used to assess the association between alleles and traits. So we'll just go over the steps to the chi-square test. So step 1 would be to formulate a hypothesis. So you have the null hypothesis, where there is no significant difference between the observed and expected genetic ratios. Any deviations are due to random chance. And we have our alternative hypothesis. There is a significant difference between the observed and expected genetic ratios, suggesting non-random genetic associations or deviations. Step 2 would be to collect the data. Step 3 would be to establish their expected frequencies. So the EF, the expected frequency, is equal to the total sample size times the expected proportion for that category. Step 4 would be to calculate the chi-squared statistic. Step 5 would be to determine the degrees of freedom. Degrees of freedom is equal to the numbers of categories minus 1. Step 6 would be to set a significance level. Step 7 would be to calculate the critical value or your p-value. And then step 8 would be to compare the chi-squared value to the critical value or p-value. That way you'll know if you reject the null hypothesis or you fail to reject the null hypothesis. It's important to know that the chi-square test assumes certain conditions, such as random sampling and sufficiently large sample sizes for valid results. Additionally, chi-square tests are commonly used in genetics, but other statistical tests may be more appropriate for specific genetic data analysis scenarios. So we just talked about how to use the chi-square test statistically, but let's tie it into real-life examples. So number 1 would, of course, be Mendelian genetics. Assessing Mendelian inheritance patterns. The chi-square test helps to determine whether observed genetic ratios, like, for example, in dihybrid crosses, match the expected Mendelian ratios. Number 2 would be population genetics. That would be using the Hardy-Weinberg equilibrium equation. Geneticists use the chi-square test to determine whether a population's genotype frequency are consistent with the predictions from the Hardy-Weinberg equilibrium equation, which provides insights into factors like mutation, migration, and selection processes. Number 3 would be linkage analysis. Investigating gene linkage, researchers can use chi-square tests to assess whether 2 or more genes are linked on the same chromosome or segregated independently. Number 4 is seen as association studies. So in genome-wide association studies, the chi-square test is employed to identify associations between specific gene variants and disease or traits. Number 5, of course, would be genetic disorders. Geneticists use the chi-square test to examine whether observed ratios are affected or unaffected individuals in families or populations match expected ratios for genetic disorders, like cystic fibrosis or sickle cell anemia. Now we can also use the chi-square test in gene expression. It can be applied to assess whether the distribution of expressed and non-expressed genes in different conditions, healthy versus disease, is statistically significant. Lastly, we can use the chi-square test to measure allele frequencies. It can be compared and observed allele frequencies in populations to expected frequencies, helping study genetic diversity and evolution.